Good day, everyone. We're going to go ahead and get started. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix. Today our webcast is presented by Ashley Hintz, our Field Application Scientist. Ashley will discuss using the RNA-Seq functionality in SVS and provide demonstration using public data. Ashley, I'll hand it over to you.
All right, great. Thanks, Cheryl. As Cheryl mentioned, today I'll be taking you through an introduction of RNA-Seq, how the data is generated, and then continue through an analysis of public RNA-Seq data from mice. If you have questions during the presentation, please enter them into the GoToMeeting window, and we will answer as many as we can at the end of the presentation. For those of you unfamiliar with Golden Helix, our flagship product is SNP and Variation Suite, or SVS. The performance, usability, and visualizations have made this software an obvious and popular choice for genetic researchers and has resulted in hundreds of citations in peer-reviewed articles. You can see a list of these articles on our website at goldenhelix.com. The agenda for today will be an RNA-Seq introduction. I will go through the data set downloaded from Gene Expression Omnibus. We'll go through a demonstration in SVS and end with conclusions. RNA-Seq workflows usually go through 3 steps: primary analysis, secondary analysis, and tertiary analysis, the sense-making step. Primary analysis is the generation of data, on-machine real-time statistics, the production of sequence reads and quality scores, and the typical end product is a FASTQ file. The secondary analysis step is recalibrating, decoupling, quality assurance, and clipping and filtering of the sequence reads. Also alignment and assembly of the reads. This can also include read quantification by gene or transcript, and the typical end product is a BAM file and or count data. And just as a side note, some desktop sequencers often have secondary analysis bundled with them, such as the MySeq or Proton. And finally, in the tertiary analysis, involves quality assurance and filtering of count data, visualization of sequence data in genomic context, sample and population summary statistics, and differential expression analysis. So what is RNA-Seq? RNA-Seq uses next-generation sequencing technology to capture a snapshot of RNA presence and quantity from the genome at a given moment of time, also known as the transcriptome. And what are the products of a secondary analysis pipeline? This can be counts, which is simply the number of reads overlapping a given feature in the genome, such as a gene, or RPKMs, which is reads per kilobase of exon per million. This term was developed before paired-in sequencing techniques, and counting reads in this way would effectively double the number of sequenced molecules, which is where fragments per kilobase of exon per million comes in. The term fragment is used to accommodate this paired-in nature of sequencing. They are normalized by dividing the total length of all exons in the gene or transcript. And finally, TPMs, or transcripts per million, is another normalization which essentially states that out of 1 million transcripts found in a cell, how many would be from this gene. We'll look at how each of these quantifications affect the results of DE-seq analysis in SVS. So now I'll tell you about the data set that I'll be using as an example today. Again, it was downloaded from Gene Expression Omnibus, or GEO. It was published this month by Mattia et al. With the accession number provided. This study looks at spinal muscular atrophy caused by a mutation in SMN1 or SMN2. This is a ubiquitously expressed gene, so RNA-Seq technology is used to study the differential expression between a case and control group of mice. This is a flowchart of the workflow that was used to generate the counts. Files were downloaded from Geo as SRA files. They were converted to FASTQ files, and then they were aligned in either bowtie or top hat. And then the freeware programs RSEM or cufflinks were used to generate output. And both alignments were based on the NCBI 37 genome with ensemble transcripts. A little bit more about the data set. Again, this is a case control study with 3 individuals in each group. The cases were mice knocked out for the gene SMN, which causes the spinal muscular atrophy phenotype, which are called the A2 group. And then 3 control mice were used for comparison, known as the HB9 group. For the demonstration today, I'll walk you through how to import RNA-Seq data in SVS. We'll do some simple sample statistics, perform a PCA, association testing, and then DE-seq analysis with counts and FPKMs. We'll also visualize heat maps and results in Genome Browse. So let's jump into SVS. For those of you unfamiliar, this is our project view in SVS. It has 3 frames: the Project Navigator window, the Node Changelog, and User Notes section. The Node Changelog keeps track of all of the functions applied to the spreadsheets, which are shown in a hierarchical fashion in the Project Navigator window, which as I go through the analysis will be evident. I already have my spreadsheets of data imported into SVS, but I will show you our import menu. We can handle text, third-party, and TPED-BED formats along with many others, but for importing RNA-Seq data, the most likely options that you'll be using are the text or third-party options. And if I open one of these, you can see for file formats we handle comma delimited all the way to tab delimited, depending on how your file is formatted. And one other thing I'll mention under the advanced options, we can also accept genotypic data as a text file, but since RNA-Seq data is numeric, you'll want to uncheck that option when importing the values. And as I stated, I have already done this for my data set, so I'm going to go ahead and cancel this and open my spreadsheet. A couple things to note about our spreadsheet format. Up in the upper right-hand corner is the dimensions of the spreadsheet. So I have my 6 samples with 36,000 markers. And then over on the left-hand side of the spreadsheet, we have this green map button. And if I click on that, it gives me the mapping information for each one of the markers with chromosome start position and stop position, which comes in when viewing results in Genome Browse. We'll also notice the blue letters labeled on each column. The C stands for categorical, B is binary, R is real number. And that just identifies the type of data in that column. So now that I have my data in SVS, the first thing I'd like to see is sample statistics. Up in our numeric menu, I can run statistics per row and leave the defaults. And the major thing I want to see from this is the sum. For running principal components analysis and association testing, we'd like a more normal distribution of the data, and I can see from the sum of the reads that my data is not normally distributed. So I can come into the RNA-Seq menu to normalize and log transform my data, and we'll leave the defaults. Okay. Close these additional spreadsheets here. This is the one I'm interested in. This is the upper quartile normalized and log transformed data. And from here, I can perform a principal components analysis. So under my numeric menu, I can run a numeric principal component. And these defaults look good, so we'll go ahead and click Run. Okay, it gives me two spreadsheets as output. I have one with my 5 eigenvalues, and then another spreadsheet of my eigenvectors. And that's the spreadsheet I'm going to look at. For visualization, I'm going to merge this with my phenotype spreadsheet. Real quick, there we are. And now I can create a scatter plot of the top 2 principal components. Let me make that a little bit bigger. And I can also make these points a little bit bigger because they're hard to see. There we are. And I'd also like to change the color. Instead of having them uniformly, I'm going to select a variable, which is my phenotype that I merged with the spreadsheet. Now I can see each of my groups they're separated into two different groups, which is explained by the principal components here. I have my A2 group being the cases and the HB9 group being the controls. And you can see in my Project Navigator window that I have a hierarchy of all of those spreadsheets that I've just created. And if I open where I've done work with my FPKM values, I've run a principal components in the same fashion with that count data. And you can see again that the two groups are explained by the top 2 principal components. So the next test I'd like to perform is an association test. So I'll come back to my normalized and transformed data and select my binary case control variable. I've just clicked on it once and it's turned this pink color, identifying it as the dependent variable. And I can come up to my numeric menu for a numeric association test. And we'll just run a t-test with the defaults. Okay, so now I have a spreadsheet of my association test results, which I'm going to set aside for now so I can perform a DE-Seq analysis, and we'll come back to the association test results momentarily. Now for running a DE-Seq analysis, it has an internal normalization, so I'm going to go back to my original count data. And under my RNA-Seq menu, DE-Seq analysis. Whoops. And it's telling me I forgot to select my dependent variable, of course. So I'll go ahead and click that. And again, I just clicked it once to turn it pink. Now I can run my DE-Seq analysis. And we'll get a little bit extra output. And click OK. When this analysis is done, we'll look at the results in Genome Browse and compare it to the FPKM DE-Seq results. And we're almost done. Okay. So I have a spreadsheet here of my association test result, my DE-Seq analysis results. And to look at my negative log p-values in Genome Browse, I can right-click and say Plot Variable in Genome Browse. And now I have those values in genomic context. I can also add in my results from the DE-Seq from FPKM that I ran earlier. And we can see immediately that they are not particularly similar. And I'm going to go ahead and zoom in on one of the highly significant results from the FPKM, which I'm going to go ahead and rename these so we know what we're looking at. FPKM. We'll edit this title real quick. Counts. Okay. So just using the scroll wheel on my mouse, I'm scrolling up to zoom in. And one more thing I'm going to add is from public annotations, is the ensemble gene track, because that's what this data was aligned to. And I can see that I have a gene here that's quite short. That's indicated as highly significant from the FPKM values, but not significant when looking at count data. And this is because FPKM values are normalized by the size of the gene. So it becomes spuriously significant when in fact it is not implicated in this phenotype. From the research that the authors did, one gene that they did identify as associated with the phenotype is NKX2-2, and I can search for that and jump to that gene. And I can see immediately that it was considered highly significant from the count DE-Seq analysis, but not significant in the FPKM analysis. And you can see that the length of this gene is quite long compared to the other. And if I place a mouse anchor here and zoom out, we can see from the value counts that it was considered highly significant across the genome. So I'm going to go ahead and turn off the plot for the FPKM values. And to continue exploring this, I'm going to add in a heat map from my project. We'll get the original count data, heat map. And I can also group these samples by their case control phenotype and zoom back into that gene. Which we can see is differentially expressed between the two groups. And if I click on that plot, let me clear my mouse anchor. There we are. If I click on each one of these samples from my case group versus my control group, I can see the values in one group are 1,000, and if I click on the other group, they're quite low. There we are. So in my control group, I have high expression values, while in the case group, I have low expression values. And I can check that again by looking at the BAM files, which I have on my computer. So I'm just going to grab one from each group. You could potentially look at all 6, but for today I'm just going to look at 2. And I can see immediately from the pileup of the BAM files that there is deep coverage for the control group and low coverage for the case group. Okay, from here I'm going to take you back into SVS because I told you we look at the association test results. And we're going to do that by merging my DE-Seq results from the count data with the association test results from FPKM. And I'm going to make a scatter plot. Okay. Now I've plotted the negative log 10 p-values from the count DE-Seq with the t-test negative log 10 p-values from the FPKM association. And I've done this to show that the markers that are significant in one test are also significant in the other test. So they're giving similar results. Clean up my workspace here, and we'll go ahead and save my project. And let's go back to PowerPoint. One more plot I can show you in SVS are volcano plots. The one on the left was generated from the count data that we generated, and then the one on the right is the negative log 10 p-values and fold change provided by the authors that we simply downloaded. And you can see on the plot on the right that there is a cutoff at negative log 10 16, which is an artifact of the program that they use to generate their p-values, while as SVS doesn't have a limit. And I've circled one of the points here which indicates the NKX gene that I showed you previously. And I can show you how to generate those plots in SVS. So I've opened my spreadsheet of DE-Seq results, and we'll plot the fold change versus the p-values. And the other thing that I did to create that other graph was to color it by a variable. In this case, I'm going to color it by p-value, and I'll make a cutoff of 0.05. So now we can see down at the bottom, the points in green have a p-value greater than 0.05, where all of the blue markers are less than or equal to 0.05. And if I click on these points, I get the information for it in my data console. And if I click on that value, it takes me to the spreadsheet. And then I can look at my map to see it where it's marking in the genome. And this happens to be the NKX gene that I showed you earlier on chromosome 2. So in conclusion, DE-Seq in SVS is best performed with count data. FPKM and TPM data can be analyzed with association testing, but is not ideal for DE-Seq in SVS. TPM value counts work the same as FPKM value counts. So with that, I would like to thank you all for watching today. Hopefully you saw something useful that can help you in your own research. And now I will pass it back to Cheryl for announcements and questions.
Thank you so much, Ashley. As Ashley mentioned, we will take questions. If you have them, go ahead and answer them into the GoToMeeting question panel. Our webcast recording and slides should be up on our website tomorrow at the earliest. Our next webcast is coming up Wednesday, October 1st, and invitations did go out this morning. This will be a very exciting one in which we unveil the newest addition to the Golden Helix software suite, which is VARSeq. VARSeq is a new tool that we will launch right after ASHG, and it supports clinical testing, allowing our users to build gene panels and whole exome and whole genome workflows in a very simple way. We hope that you can join us. That's all for me, so back to Ashley for some questions.
Okay, so it looks like we have some questions today that are very statistically intensive, so I'd like to confer with our statistical geneticist before answering, but rest assured your questions will get answered by the end of the week.
Okay, thank you so much, Ashley. Thank you, everyone, for attending. We're going to go ahead and wrap today, and we will see you next Wednesday, October the 1st. Have a great day.